Good morning, everybody. Thank you for being here. We got a good crowd. People are going to keep coming in probably for a few minutes, but we'll get started anyway. My name is David Carr. I work for the Finger Lakes Institute at Hobart William Smith Colleges in Geneva, New York. I manage the Starry Stonewort Collaborative. Most of you probably knew that already. A couple of housekeeping things. So most of us, or many of us at least, are still working at home. So put your mutes on if you would, please. Mute everything. We're all home and we still have potential for dogs and fire engines and who knows what else making noise in the background. So for everybody's sake, let's keep mute on. And as soon as I'm done talking, I'll mute myself. This is our first, last year we did a similar thing. We had 4 summer webinars, one each month during the summer. And we're doing the same thing now. And we're lucky to have Tyler Harrow-Lyle here to kick us off this year. Tyler is with Ontario Tech University in Oshawa, Ontario, which is, I guess, just east of Toronto along the Lake Ontario shoreline. He's a PhD student. He's in the Applied Bioscience program. He's working with Dr. Andrea Kirkwood, who is also on the call today. Thank you, Andrea, for being here. And his title is Investigating the Distribution of Starry Stonewort in Ontario Lakes and its role as an ecosystem engineer, which sounds really interesting. Because as many of us know already, this stuff can move in and just kind of take over. So in this research, he's focusing on investigating physical and chemical habitat characteristics that promote and constrain Starry Stonewort's invasion of Ontario lakes. It also includes determining potential impacts on communities present through the lower aquatic food web in the environment. So I think that's it. Oh, keep mute on. We've got a good crowd here today. So if you have questions, please put them in the chat box and we will address them at the end. Either I will read them out or Tyler will read them from the chat box. I think we're gonna have too many people to have an open conversation. So thank you for that. So with that, I think Tyler, go ahead and share your screen and start when you're ready.
Okay. So thanks for everyone for coming out today. And like David said, I'm going to talk to you about investigating the distribution of Starry Stonewort in Ontario lakes and its role as an ecosystem engineer. So despite looking like an aquatic plant or a macrophyte, Starry Stonewort is actually a green alga, a part of the Coriaceae family. Coriaceae are known as stoneworts or brittleworts, and there are over 800 species worldwide. Unfortunately, the majority of the stoneworts are now classified as endangered due to increasing human pressures. Panel A demonstrates what a typical rake collection can look like in systems where Starry Stonewort is the dominant macrophyte. You can see it can become the only visible macrophyte present on the rake, but when we take it back into the lab, we often find that several other species are commingled in. If we compare panels B and C, you can see that Starry Stonewort can change what it looks like based on environmental conditions. Both of these samples were taken from Lake Simcoe in relatively close proximity to one another. Traditionally, without prior knowledge or experience with Ontario stonewort populations, panel B would be classified as a Cara species. And this follows a history of misidentifications throughout the native and invaded ranges. Correctly differentiating between the native stoneworts of Ontario is extremely difficult, as we have such a diverse underlying geology, which we will talk about more in depth shortly. Other examples of misidentification also come from the native range within Japan. In Japan, they thought Starry Stonewort was extinct until research using DNA analyses revealed that Starry Stonewort was still there, but it changed its appearance due to increasing environmental pressures. In Ontario, only males or specimens that do not produce sexual organs have been observed so far. Panel D demonstrates what the male sexual organ looks like. They're also called antheridae. They're these little tiny red spheres. These can be distinguished from female sexual organs, as those would have a spiral jacket and a crown-like cap on them, but they would also appear as little red spheres. Starry Stonewort can displace all other macrophytes in Ontario systems where it becomes established, as we can see in panel E. The common name of Starry Stonewort comes from the association with conspicuous white star-shaped bulbils, as we can see in panel F there. And these bulbils serve as asexual reproductive structures and hibernation cells. These arise from the clear monofilament at the base of the alga, which we can see as well in panel F. And this monofilament acts as an anchor in cases where Starry Stonewort is attached to the sediments. Although bulbils can help in identifying Starry Stonewort, they can prove unreliable for early invasion fronts, as with Ontario. Ontario appears to be 10 years behind the invasion of lakes as seen in the United States, and bulbils are only produced within established beds of vegetation. Often bulbils of other charophytes are frequently mistaken for those of Starry Stonewort within Ontario, which further confounds positive identification. Starry Stonewort is native to Europe and Asia, stretching from the British Isles to Japan, and throughout most of its range, it's classified as an endangered species. In North America, known distributions have been recorded for central eastern portions, and the earliest record was observed in 1974 along the St. Lawrence River. Despite extensive reports from invaded regions in the United States, prior to our research, Starry Stonewort was only confirmed within 2 systems of Ontario. Starry Stonewort was found in Lake Simcoe by Dr. Brian Ginn as early as 2008, and within Presqu'île Bay of Lake Ontario in 2015 by Dr. John Midwood. Our work on Starry Stonewort originated from a research partnership with the Scugog Lake Stewards. In 2016, the Stewards initiated this partnership with Dr. Andrea Kirkwood's lab to investigate population collapses they had not previously seen. We identified Starry Stonewort in 2016, and for the following 4 years, we investigated the effects that it was exerting on the environment and communities across the lower aquatic foodweb. And we wanted to sort of use Lake Scugog as a sentinel ecosystem to begin to understand what was to follow in the invasion of Ontario lakes. Although Lake Scugog has long been considered a weedy lake, which is a nuisance for recreational uses and watercraft, historically, Lake Scugog has been an extremely productive system. As a result, the regional economy gained $10 to $15 million annually by tourism and recreational activities supported by the lake. In recent years, Lake Scugog has faced increasing threats to ecosystem health by multiple stressors, including climate change, land use activities, and invasive species. Lake Scugog has faced many invasive species, but the most notable are Eurasian water milfoil, pictured on the left, and zebra mussels on the right. Eurasian water milfoil has been referred to as the most aggressive invader to be introduced into North America, but we'll shortly talk about why this is not true for Ontario. Native to Eurasia, Eurasian water milfoil was introduced to North America and has been present in Lake Scugog since the 1960s. Zebra mussels are also native to Eurasia and were identified in Lake Scugog in 1991, though their numbers remained relatively low until recently. Despite Eurasian water milfoil being labeled as the most aggressive macrophyte introduced into North America, Starry Stonewort easily displaces it, along with everything else. Upon our initial community surveys in 2016, we observed Starry Stonewort and Eurasian water milfoil co-occurring within our samples. However, as we continued our surveys, we saw Starry Stonewort was displacing, or in some cases, completely eliminating Eurasian water milfoil populations. This was interesting because previously, Eurasian water milfoil was thought to have detrimental impacts on aquatic communities that were present. However, using richness analysis, we actually saw that Eurasian water milfoil populations were beneficial for the ecosystem, where Starry Stonewort completely eliminated almost everything. In addition to the population collapses we observed, 2016 was the first year where widespread phytoplankton blooms erupted in Lake Scugog. Up to this point, there were no historical reports for phytoplankton blooms in Lake Scugog. These blooms were dominated by the toxin-producing Taxa microcystis. This has detrimental impacts at an ecosystem level, but can also become problematic for human health in the coming years, as surrounding municipalities rely on Lake Scugog as a source for drinking water. Yet, despite the increase of Starry Stonewort and the emergence of microcystis blooms, other problematic taxa seem to thrive. Throughout our 4-year study, we tended to see an association between Starry Stonewort and microcystis, as we can see on the graph on the left. Generally, where we saw higher biomass of Starry Stonewort, we would also observe higher concentrations of microcystis, often resulting in a localized bloom. Similarly, with higher abundances of Starry Stonewort, we saw an increasing density of zebra mussels. If you recall, Zebra mussels have been in Lake Scugog since 1991, but their numbers remain relatively low. Since Starry Stonewort has a hard marl incrustation on its body, this acts as a perfect substrate for zebra mussels to attach to. This association can be particularly detrimental to an ecosystem, as zebra mussels have been noted to further promote microcystis blooms in other systems through various mechanisms. This led us to question whether the negative things we were observing was a product of invasional meltdown. Invasional meltdown is a term used to describe when negative effects of one invasive species are exacerbated by another. To investigate this idea, we created an ecosystem model to identify what was driving microcystis blooms within Lake Scugog. Historically, bloom drivers are known to include things such as phosphorus concentrations, precipitation, and chloride. We observed that precipitation and chloride were important drivers for microbloom formation within Lake Scugog. But for the first time, we identified Starry Stonewort as a direct cause for these emerging blooms. It's also important to note that regardless of the history zebra mussels had in supporting microcystis blooms in other systems, we demonstrated that this was not the case for Lake Scugog. The only biological interaction with a meaningful relationship for these emerging blooms was between Starry Stonewort and the microcystis. Even though we did not see phosphorus playing a limiting role or play an important role in the emerging microcystis blooms, this is likely due to phosphorus being in excess concentrations and not limiting microcystis within Lake Scugog. We were also able to quantify how Starry Stonewort was affecting phosphorus concentrations within Lake Scugog, allowing the perfect bloom opportunities to occur. An essential part of Starry Stonewort's reproduction cycle includes low oxygen concentrations at the sediments. In order for Starry Stonewort to reproduce from bovials or other asexual calcified structures, often called gyrogonites, deposited within the sediments, low oxygen concentrations and a fraction of available light must be present. When these things occur, the reproduction cycle is initiated. We observe sites with Starry Stonewort to have lower oxygen concentrations at the sediments, suggesting Starry Stonewort is using this bubble bank within the sediments as a reproduction mode. Furthermore, it is not uncommon for the sediments to experience low oxygen concentrations in very productive systems, such as Lake Scugog at the peak of the growing season. But Lake Scugog is classified as a polymictic system, which means due to the relatively shallow depth, winds, and other forms of turbulence, there's continual mixing, so this generally does not occur. Despite this, we still observed a biomass response and in oxygen concentrations, where we had more Starry Stonewort oxygen concentrations were becoming depleted regardless of the system being polymictic. This can be problematic as when oxygen approaches very low levels at the sediments for prolonged periods of time, the dynamics for nutrients such as phosphorus change. Generally, phosphorus is removed from the water column as it binds to particles and settles into the sediment. But when the change in nutrient dynamics occur at the sediments, phosphorus can be released back into the ecosystem, termed as internal loading. With increasing biomass of Starry Stonewort, we saw oxygen concentrations approach these very low levels, resulting in the release of phosphorus in a biomass-dependent manner. For Starry Stonewort facilitating internal loading of phosphorus, it indirectly contributed to furthering the perfect bloom conditions we were observing within Lake Scugog. From our work in Lake Scugog, a sentinel system for Ontario, we had identified how Starry Stonewort was acting as an ecosystem engineer. Within an ecosystem, Starry Stonewort is decreasing diversity across the aquatic communities. It is also altering the sediments, resulting in changing water quality, which has further impacts for the aquatic communities that are present. Despite our understanding for how Starry Stonewort was acting as an ecosystem engineer within Ontario, we did not fully understand what habitat characteristics either promote or constrain the invasion, and whether the diverse landscape of Ontario had any influence on this whatsoever. The overall distribution was also not known outside of the two previous reports and our now confirmed presence within Lake Scugog. So to begin to address these gaps in knowledge, we surveyed 60 lakes across the 65,000 square kilometer area of Ontario. In Ontario, we have a unique geological feature known as the land between, where a shift in parent geology is present, going from limestone dominated in the south to granite dominated in the north. This gave us the perfect opportunity to identify the habitat characteristics that promote and constrain Starry Stonewort's invasion of Ontario, with particular emphasis on a naturally occurring cation gradient. One notable cation included was calcium. Calcium has very important biological roles for Starry Stonewort, where it cannot persist in environments with concentrations below 15 milligrams per liter. We expected to see a strong concentration response within our candidate lakes as calcium increased, and this is similar to what has been observed within invaded regions of the United States. Our candidate lakes range from 0.4 to 63.8 milligrams per liter, and this was split between three categories. As stated almost 100 years ago, the minimum threshold for calcium for Starry Stonewort to persist beyond transient populations remains between 15 and 25 milligrams per liter, depending on other environmental conditions. There's also been suggestions that the optimal range for establishment occurs between 20 and 40 milligrams per liter. Our candidate lakes were split 20 per category, where 0 to 20 represented below threshold, 20 to 40 represented the optimal calcium conditions, and 40 plus represented above this threshold. So across Ontario, we documented Starry Stonewort in 19 of the 60 study lakes at 37 sites. The black dots here represent lakes where Starry Stonewort was identified, and the gray dots correspond to the other candidate lakes where Starry Stonewort populations were not found. The underlying colors correspond to an extremely simplified version of the underlying geology within Ontario that ultimately influences the cations within each of our candidate lakes. 11 of these lakes did not have macrophyte communities present at the sampling locations when we were out sampling. And Starry Stonewort populations were found to stretch from Brockville, a city between Kingston and Ottawa, and Eugenia, which is northwest of Toronto, near Georgian Bay of Lake Huron. Our recorded distribution is vastly larger than what has previously been reported for Ontario, and this is likely due to inadequate invasive species monitoring, as aquatic systems are not monitored in Ontario until drastic ecosystem changes are observed. After our study concluded, several unconfirmed reports emerged for Starry Stonewort for the same area that we investigated. These reports claimed a much higher distribution than what we actually detected for the area. However, given the abundant native stonewort populations and diverse environmental influences present that can result in Starry Stonewort altering its appearance, these reports require investigation and confirmation, likely within a lab. Starry Stonewort is a cryptic invader. Cryptic in this sense uses a more zoology-based definition, where an organism is basically tries to hide itself or uses its surroundings and other organisms to survive or evade detection. As I mentioned before, stoneworts can change their appearance based on environmental conditions of an ecosystem. Across native and invaded regions, researchers dedicated to studying stoneworts have noted high degrees of misidentification attributed to this fact. And across our study, after accounting for the environmental influences, we observed strong positive associations between Starry Stonewort and native stoneworts within Ontario. At a quick glance, it is extremely difficult to differentiate between these algae, with subtle differences becoming obvious when specimens are taken back to the lab. Further examples of the history for misidentifying the species have been encountered throughout our research as well. Occasionally, we invite research partners to the lab to educate them on methods for differentiating between these local populations, and this revealed that Starry Stonewort was misidentified for several years prior. The most common taxa for Starry Stonewort to be confused with is Nutella, and across Ontario, Starry Stonewort and Nutella were found to have strong co-occurrences with one another. Unfortunately, generalized identification keys often fail to be widely applicable for Ontario stonewort populations, as they are in other regions, which further complicates conservation efforts and initiatives. We can further see this by browsing reports of Starry Stonewort invasion from third-party reporting applications. Stonewort meadows, like you can see on the left here, are extremely abundant in Ontario lakes. Several reports from one of our candidate lakes that occurred within the same month and relatively same geographic location emerged, claiming Starry Stonewort populations were present. Yet when we did our synoptic survey, we failed to confirm the presence of Starry Stonewort. The stonewort present was not Starry Stonewort, rather Nutella, a taxon commonly mistaken for Starry Stonewort. And unfortunately, despite several Starry Stonewort initiatives occurring within Ontario, the complexity of the underlying geology and the innate ability to alter the appearance based on environmental conditions are very confounding for initiatives going forward. Earlier, I mentioned that calcium is the essential driving factor for Starry Stonewort invasion within America. And in invaded regions in the United States, Starry Stonewort responded with increasing calcium concentrations. In contrast, we observe Starry Stonewort occurring in a much more constrained range, which ranged from 15 to 57.9 or 8, milligrams per liter. The black dots here represent where Starry Stonewort was found along our candidate lake calcium gradient. And as you can see, they generally tended to stay more clustered around the lower end of this range. So to better understand these differences and identify the environmental constraints for Starry Stonewort in Ontario, we created an ecological niche model. What this model revealed was that other parameters were stronger predictors for Starry Stonewort's invasional success compared to calcium. Calcium was still an essential component for Starry Stonewort presence, but other factors like potassium and magnesium concentrations were better predictors. We also observed that higher phosphorus concentrations tended to coincide with Starry Stonewort presence. Interestingly, we found that high concentrations of manganese had an inhibitory effect on Starry Stonewort presence across Ontario. Although this has not been reported for Starry Stonewort previously, this is a known effect on other stoneworts. Depth was the strongest predictor within our model, where Starry Stonewort tended to occur at shallow sites closer to boat launches. This follows work that came from invaded regions of the United States and suggests the invasional front within Ontario is still very early, likely 10 years or so behind what is observed in the United States. It's also important to note that our ecological niche model identified several lakes where we did not detect Starry Stonewort populations, even though the habitat characteristics were ideal for populations to exist. And we hope that various stakeholders apply our ecological niche model to long-term monitoring data that's somewhat widely accessible throughout Ontario to identify at-risk ecosystems and direct management and implementation as that becomes available in the coming years. So in conclusion, Starry Stonewort is much more established than previous reports. However, the association between Starry Stonewort and other members of the Stonewort family are going to further confound our identification efforts and create more inaccuracies. Starry Stonewort is acting as an ecosystem engineer by altering sediment dynamics and facilitating population collapses across aquatic communities. Starry Stonewort is also causing the increase of other problematic taxa, which exacerbate the negative effects on the ecosystem and communities. Our ecological niche model revealed other cations are better predictors for Starry Stonewort presence in Ontario lakes, but calcium is still an essential and required component. Populations of Starry Stonewort have yet to be identified within ecosystems, which from our ecological niche model, which highlighted the full distribution for Starry Stonewort is not currently realized. And with the proper enforcement and increased awareness and management implementation, there's still hope to preserve most of Ontario's at-risk lakes. So with that, I'd like to thank everyone from the Kirkwood Lab, our research and funding partners, and I'd be happy to take questions.
Tyler, thank you very much. That was excellent, really good, and a little unsettling. Some of the relationships between Starry and microsystems and some of the other things are, boy, not good, but we're glad you're out there doing it and figuring these things out. This is a really excellent research. We've got a couple questions I'll start with, and if anybody has any other questions, please put them in the chat box and we'll keep going here. Carol asks, what other Cara produces bulbils? Tyler, we lost your audio, Tyler.
Sorry, can you hear me now?
Yep.
Okay. Pretty much every other type of stonewort has the capability to produce bulbils in Ontario, although they're not always present. For other charophytes, bulbil production generally is determined by environmental factors. For Starry Stonewort, in early establishment, bulbils will not be produced just because they don't have the capacity to do that yet. But for other stoneworts, pretty much everything has the capability for bulbil production.
All right, we have a comment here from Christine. There's a large presence of Starry Stonewort across the Bay of Quinte. I hope I'm pronouncing it correctly, in Lake Ontario. It says we have wetlands that have been routinely monitored since 2006 for macrophytes.
Yeah, I'm not sure if this is in relationship to the work that Dr. John Midwood did, but he was in that area and they I believe it was 2016, published a paper where they found Starry Stonewort all throughout that area in the wetlands of Lake Ontario. That's good to know that they've been monitoring that since 2006.
Yeah, it's excellent. Here's the one from Tully Lake, which is east of us, about an hour east of us here in Stringy Lakes. Tully Lake is Cara and Starry Stonewort, which also looks very similar. It's kicking our butts, in parentheses, more HABs along with the Eurasian milfoil. And maybe chemical mitigation in the future, question mark. Thanks for this presentation.
So this is just my personal opinion, and it might differ from other experts in the field, but chemical and physical management of Starry Stonewort is largely not successful at all. Just based on the life history and reproduction cycle of stoneworts in general, if you apply a widely applicable herbicide, you kill everything off, and stoneworts are considered rapid colonizers, so they'll actually increase their population presence if you do that. Same with physical removal. If you remove the competition, stoneworts can very rapidly reproduce and just take over the area. So going forward, management, you have to be very careful not to cause more harm and be contradictory to our original purpose. But I'm very hopeful. Like, for instance, so far in Ontario, we only observe male individuals or specimens that are considered sterile, which means they don't produce sexual organs. So we're actually working on a separate project for DNA analysis where we hope to identify whether it is only males present. Because if we identify if a population within Ontario is only producing clonally, this often opens up a new management perspective, so we can investigate potential methods to implement.
Thanks. That leads into the next question, which you kind of just answered. What is the latest in eradication strategy?
I know within the United States, they often apply herbicide or do physical removal. Even in like Scugog, they try and harvest using like physical removal. I'm not sure what the machine is. It's just a giant machine thing. I often say that we shouldn't be doing that right now. We simply don't know enough about the invasion biology and overall life history traits. We tend to try and reinvent the wheel when it comes to stuff like this, where if we just look at the literature from 100 years ago, we would realize that such management opportunities or practices that have been implemented simply will not work. It's like pouring gasoline on the fire.
Follow on. So, Greg asks, are the other stonewort bulbil star-shaped as well?
Sorry, you cut it a bit. Could you?
Are the other stonewort bulbils star-shaped as well?
Yes and no. So, like, stoneworts in general can change their appearance, both as on their body, the axis and thyli, as well as bulbils based on environmental conditions. In some situations, even I was confused because sometimes on Cara specifically, I think one case in Nutella, you can see the bulbil start to have a little outjet, but when you actually examine it under the microscope, you can see vast differences. So that can be sort of confusing. I'm not sure if that's widespread or very specific for Ontario, but like I've been confused a few times, and I actually had to reach out to other researchers for aid in this because it's not always clear cut. Even with the bulbil presence, it's sometimes very confusing where when you take it back to the lab and actually process the sample, that's when it becomes evident.
Interesting. Christine, follow up from Quint Bay. We also have a long-term monitoring program that monitors for microsystems in algal communities. And yes, John Midland report came from our area. Barbara, why might Starry Stonewort be only male? What would happen if it wanted to develop a male and female? And also secondarily, what is Starry Stonewort relation to fish populations?
Okay, so stoneworts in general are somewhat unique. Male individuals are produced first and earlier in the growing season. And as the season progresses, that's when females would emerge. If we look at the native range, female populations are extremely rare there as well, even though generally in a shallow lake, I'll use Lake Scugog as an example, that's generally where you would see a shift from the asexual only males present to sexual reproduction. But there's been low fertility rates and low sexual reproduction in Starry Stonewort present for, I think it was I think the paper was 1937 as early, with earlier reports. So if it ends up developing both male and female, that just means that the growing season, like it totally has the capability to do that. But being outside of the native range, we're not quite sure yet whether females are actually present. They haven't been found yet. And this in part is due to we're not quite sure whether the climate and environmental conditions are optimal for females to develop if they develop later in this growing season. And for fish populations, no quantitative research has been done on fish populations. I know we've tried to talk about this previously. I don't know about other invaded regions, but within Ontario, doing fish populations, you need a whole bunch of permits and stuff. So the work that we basically do for fish populations is inference-based. We look at the habitat quality. You can't have a healthy fish population if, for say, there's no community heterogeneity, which means a very diverse population. If you have one species which completely pushes everything else, causes anoxia, promotes microcystis blooms, we can infer that fish populations will be negatively impacted. I think there's one instance from, I believe it's Michigan, by Dr. Pullman, where he observed that some fish populations were actually using portions of the Starry Stonewort meadow as a nest, but these were also invasive species of fish, if my memory serves me correctly.
Okay, Michelle asked about full citations for the articles you've cited, and Andrea Kirkwood has very quickly posted those in the chat for everybody who wants to review. Okay. And she also says, we are happy to send you a PDF copy. Just send Tyler an email, and your email is there. Thank you for that. Doug asks, is it fair to say the biggest spreader of Starry Stonewort is watercraft going through it and cutting through it?
Yes. So, yeah, yes, I would agree. That's true. Stoneworts can be transferred through animals, like birds and such, but there's no quantitative research that actually investigates this within any invaded region, to my knowledge. But what we can actually see is if we, for instance, I believe it was in New York, a researcher there went to more secluded lakes, and they did not find populations of Nyctalops or Starry Stonewort there. And we also saw similar things within Ontario, where we would go to this candidate lake down some fire route, and it was extremely difficult to find, and we didn't find Starry Stonewort populations. But when we did our ecological niche model, it was ideal for Starry Stonewort invasion. So we believe that Starry Stonewort is being transported mostly through human means and watercraft. If a watercraft does go through it and cuts it, the fragmented portions can reproduce new populations. But also, if you take your boat out, and let's say in the same day or within 7 days, you go to another lake without properly cleaning it and draining it and following those protocols, you can actually introduce either like tiny pieces of baubles, little clumps and such from either your trailer or boat into a new ecosystem. And that's generally what we found, which corresponds to work from the United States, where Starry Stonewort generally tends to be in shallow sites closer to boat launches at the early stages of invasion, which suggests that boats are the main focus for transportation.
We have a very active watercraft steward program out of our office, and we post mostly college students at, I think, 20 to 25 different locations across the state, and they check thousands of boats in the summer. And we would agree that in most of the Starry Stonewort, you can find it if it's introduced. It's almost always, gathered right around the marinas and the boat launches and stuff.
Yeah, and even across our survey, most people that we encountered at boat launches don't care. They didn't watch their boat, and luckily I had a very, very good helper, Claire. She would often like go tell them, wash your boat. So,
Well, that's exactly what our watercraft stewards do here. We train them carefully, and they talk with people all summer and give them information, trying to explain the importance. And clean, drain, dry is the drum we beat here, trying to get boats cleaned off. And don't, because everybody here, like I'm sure in Canada, they fish here and then they go take the afternoon and fish in another lake too. And you want to make sure everything's clean on your boat before you travel between the lakes.
yep.
So yes, it's a major problem. Do the references include, this is sort of you and Andrea, I think, do the references include a list of all the lakes that were studied?
Yep. Yep.
She also said, are you going to add them to the Starry Stonewort monitoring database? I'm not sure what that
I'm not quite sure what that
is.
I don't know if they mean like ED, correct.
ED maps.
Yeah, or iNaturalist. I know that we've spoken about EDD or maps before. We haven't done that yet because the manuscript was in review, but in the manuscript itself, it lays out all the lakes that had the presence. Supplementary table 2 actually has all the GPS coordinates and everything for all the candidate lakes. And if you want more additional information, just send me an email and I'd be happy.
Yeah, I apologize, Tyler. I just scrolled down further. The questions Andrea said to Sheila that the lakes are listed in the second paper above, but if you message Tyler, he can send you the list directly.
Oh, okay.
So there you go. Carol asked, for monitoring programs in Ontario, do you have some suggestions for more accurate identification?
Yes. So a little, I guess, background story. Initially, I was using a lot of identification guides that were developed for the States. And when we first went out for our synoptic survey, because Lake Scugog was very, very easy to differentiate because Starry Stonewort was so established. But when we started to go to some of these lakes where it was extremely difficult to differentiate, and even what we call the Bible for Macrophytes by Wood, it was not quite as easy. So for identification in Ontario, I keep trying to suggest one unified education effort because the locally sourced populations are so different. And even within a lake, within 100 meters of one another, like we see in Lake Simcoe, like Scugog, Dalrymple, doesn't matter where we go. The environmental influences, just because of the underlying geology, are so great that there has to be a unified education to correctly identify it, so we don't further contribute to the history of misidentification and lead to more inaccuracies that could ultimately impact management implementation.
Richard asked, you mentioned the effect of starry on oxygen levels. Could you discuss that impact some more? Also, any effect of starry on phosphorus levels in lakes?
Yeah. So for oxygen, like I mentioned, low oxygen concentrations at the sediment or the benthos where the sediment meets the water is required for reproduction cycles to be initiated if Starry Stonewort is using the bulbal bank within the sediments because they can act as hibernation cells. But interestingly, Starry Stonewort can actually alter the oxygen concentrations within an environment because the hard incrustation on the body of Starry Stonewort just doesn't appear. So when they alter the oxygen concentrations, they shift the chemical dynamics in order for the phosphorus calcium bicarbonate to mix into this matrix and actually precipitate on its body. So that's another mechanism where Starry Stonewort affects the oxygen levels. For effects on phosphorus, generally stoneworts are very important players in lake biogeochemistry. And what I mean by that is they can actually alter phosphorus cycles. So like what we see in Lake Scugog, by creating anoxia or low oxygen concentrations at the sediments, they're actually changing the chemical dynamics and how phosphorus is usually processed within an ecosystem. So phosphorus is then loaded back into the ecosystem, where either Starry Stonewort can use that in creating the co-precipitation on the body, or for growth, or as we see, because Starry Stonewort has light limitation cellular mechanisms, it can also promote other problematic taxa to bloom, like microcystis.
All right, thank you. Your research again is just really eye-opening. You're doing some really interesting things. Some new things I've heard here today that I haven't heard before. I think others may agree. I think the questions have slowed down here. Does anybody else have anything else for Tyler? We've got plenty of time here. But if that's it, we're good. The weather is absolutely beautiful here. I hope it is everywhere. Maybe everybody can sneak out and enjoy it. So Tyler, thank you very much again. This is great presentation, fascinating research. Carol asks, what is next for your research?
I'm actually finishing, well, with COVID and stuff, things sort of got impacted, but I'm trying to finish this August, and I'm hoping to continue this research either through volunteer opportunities, or I'm going to start sending out some applications to try and find new research avenues so I can continue the work on Starry Stonewort, just because I'm so fascinated. But what's next is just basically we have several manuscripts in review. Dr. Kirkwood has a new undergraduate thesis student who's going to continue some of the community science-based monitoring for Kawartha conservation. So I'm going to be helping out slightly with that. But other than that, I'm just trying to continue somehow.
Fantastic. It's all good. And there's many thank yous and excellent presentation things coming through in the chats. So you very much appreciated what you did today, and your work is wonderful.
Thanks.
And Andrea mentions there's going to be some genetic work as well. That's interesting.
Oh, yeah, I forgot about that. Yeah, so when we did the synoptic survey, we actually took DNA specimens just to verify what we were seeing. It's similar to what I was talking about for Japan, where they thought that Starry Stonewort was extinct, but when they actually looked at the genetic profile, they realized it just completely changed its appearance. So we're hoping to do that, as well as determine whether only, like, be able to identify the sex of Starry Stonewort, see whether only males are present and if they're clonally reproducing, or if we just have not found populations of the females yet.
Excellent. Thank you, Tyler. Could the PowerPoint be made available?
Yeah, sure. I can, if you just want to send me an email, I can send it to you. I don't.
Yeah, can we, you could, those that want it could either email Tyler directly or me, because I also have Tyler's email address. Maybe Tyler, can you put your email again just real quick in the chat box?
Oh,
possibly, or no? Because I don't have it on the top of my head.
there we go.
Beautiful. So there's Tyler's. We'll leave this up for a couple of minutes. There's Tyler's email if you need further questions or want his PowerPoint, or talk more about the sources he cited and so on. So with that, I think we're going to wrap up. Thank you again, Tyler, very much. Thank you, everybody, for coming out to this and listening on such a beautiful day. And our next one is next month. And he's a local gentleman from a small lake here in New York State. They went through the process of New York State's somewhat legendary permitting process and did some chemical treatment for Starry Stonewort. So it's going to be interesting to get a small lake association viewpoint in that. That's in the middle of June. We'll be sending out, or actually, I'm sorry, it's early June to coincide with New York State's Invasive Species Week. So you'll be seeing flyers for that as well. And I think it was mentioned in the one that went out for this. So please join us again in June. Have a great day, everybody, and thank you.
Yeah, thanks for having me.
You're welcome. Thank you.